Hi, hello everyone, and thank you for joining us today. I'm Denis Kural, SVP of Antibody and Target Discovery here at Absci, and I'm really excited to welcome Bharath Ramsundar, the CEO of Deep Forest Sciences today. Bharath is going to be discussing the importance of open source foundations in modern computational drug discovery efforts with us today. But before we get started, just a note about questions. We encourage you to ask questions throughout the presentation. If you have a question, please press the raised hand button at the bottom of your screen, and we'll call you in real time on audio. Then a pop-up will appear asking you to unmute, which you'll need to click before we can hear you. We're recording this for distribution on YouTube. So if you'd prefer to enter your question using the Q&A window, that'll work as well, and I can just ask them out loud. So with that, I'll hand the controls over to Bharath.
Awesome. Okay, let me go ahead and dive into it. So, well, first, thank you all for kind of joining in today. Today, I'll be speaking to you a bit about, DeepChem towards open source foundations for drug discovery. Some of you may have already heard about DeepChem as a project, but for those of you who haven't, DeepChem is a framework to very broadly apply AI to scientific problems. We started life as one of the first libraries out there that provided good tools for applying graph convolutions to molecules. And since then, the community's kind of grown outwards, and I think we now have one of the most sophisticated open source scientific machine learning kind of frameworks communities out there. So DeepChem has dozens of different machine learning models. It has, again, dozens of different inbuilt data sets and scientific featurizations and transformations. And it has also been used, I think, in a very broad range of scientific papers and contributions. I think last count I did several hundred papers at the minimum that use DeepChem to do meaningful things. And actually, several organizations have used DeepChem to fruitful purpose. So I think every kind of company listed here on company or academic institution listed here has written either a paper or blog post with DeepChem or has publicly discussed their DeepChem usage. So I think we've had a pretty decent impact in terms of people actually using us to do drug discovery. And among other things, there's also nearby fields, things like designing better pesticides or herbicides or antibiotics or for environmental purposes. So there's a broad range of tools that we support kind of through the usages that we support through our open source community. We have kind of a quite active and growing community of users that have been using DeepChem to do things. One of the coolest things I think about on the project is that we have developers from across the world. I think we have active developers, India, throughout the US, throughout Europe, South America. Sorry, I have a bit of a scam call there. And Japan, among other places. So I think we have one of the largest global communities of distributed researchers kind of centered around building open source tools. And I think part of the magic of open source is I think it enables these kind of collaborations and connections that you wouldn't have made otherwise. So even if you're at a top tier institution, place like Stanford or MIT, you see people who are at that campus, but who you don't meet is the brilliant kid maybe out of Peru who would not have had a chance to come to MIT, but is by happenstance an amazing programmer. So I think that's the type of connections that DeepChem enables, and that's part of the reason I've continued to spend time on open source efforts over the years. So part of what I'm hoping to do today is one, provide just a rough introduction to what DeepChem is. And two, I'll say a little bit more about some of the science that we've enabled over the last couple of years and talk about some of the recent preprints and manuscripts that have come out of the project. So this is a very high level, a rough introduction to the structure of a DeepChem program. This is taken from our open source research forums as a diagram that was put up kind of talking through the structure of DeepChem. So DeepChem at a high level is a framework for, constructing scientific machine learning workflows where you essentially take in input data, specify a series of transformations, and then at the output get predictions or kind of other structured output that tells you, say, what molecules to try, what, sequences to look at, what images to focus on at the other side. So DeepChem allows you to kind of pick and choose every one of these components. So there's literally dozens of choices for each of the boxes you see here. So dozens of models, dozens of featurizers, different ways to split data, transform data. And when you add all these kind of combinatorial choices up, DeepChem supports a very broad range of scientific programs within a kind of simple but very flexible framework. And I think part of the simplicity here has been important for getting people onboarded, because unlike many standard scientific tool chains, you don't say need to deeply understand partial differential equations or statistical thermodynamics in order to be able to use. You can understand some basic principles of how to work with these systems and then start using that to do meaningful science, which we found is just an amazing tool to onboard newcomers to working with DeepChem.
So speaking of newcomers, in your previous slide, there seems to be an inflection point happening in early March 2020 or so. What do you think happened?
Well, there's a basic human thing there where it's like so if you can see, like when I was doing the project started, I started the project when I was doing my PhD, and it was growing quite rapidly. But then in the middle there, I decided, made a mistake of doing a startup that wasn't related to DeepChem, and continued working on DeepChem a bit when I could on nights and weekends. And I think around March 2020, kind of my time at that other company come to an end. And I thought, I'm really passionate about this. I've been spending so long working on it. Let me try to see where it can go.
So it's not the pandemic. It's not because of COVID. You had more people at home being.
It's a good choice. I think there's a kind of confounding variable there, of course, with the pandemic. I think that has had a big effect in terms of like people kind of coming online. I'll say, the pandemic triggered this amazing just wave of interest in kind of computational drug discovery. But I think one challenge still is that, like, what we saw was that there was like this spike of interest that lasted about a month. And then, after that, it kind of died down a little bit, because the reality is it's hard work, I think, in order to be able to learn these skills. And it's hard to learn enough in a month to have a meaningful impact. But we did pick up a few. I think there is a secondary kind of factor there. Yeah, so I think, of course, both those things, I think, are probably more the pandemic. Who knows?
Thank you.
So I think one of the things that we maintain as part of DeepChem is we have an extensive collection of community-driven tutorials. So these all run on Google Colab. I think there's something like now 50 tutorials that talk you through using DeepChem to solve problems in practice. So if you're doing things like well, I think we have the deepest support for really small molecules. So if you're designing small molecules, generating small molecules, looking at synthetic feasibility of small molecules, there's a tutorial in here for you. So there's a very broad range of useful tools. And off the record, I know that several companies have gotten started from people working with some of these tutorials and resources and using that to build the first version of their systems. I think I'll also mention, I'll jump ahead briefly. We have a book with O'Reilly where it's called Deep Learning for the Life Sciences. This provides kind of a more book-length introduction to working with some of the tools that are available in DeepChem. We are slowly working towards trying to get an updated version of this book out there. I think there's, of course, been a lot of fundamental advances in machine learning over the last, for the life sciences. AlphaFold 2, among other things, have really, I think, changed the face of what it means to do kind of machine learning in biology. So I think we're trying to see if we can update our resources to reflect more of that. But I will mention the tutorials are free on the internet. The book is, we don't set prices. I think it's something like $50. So most of the material in the book is covered through open tutorials as well. So if you're feeling thrifty, I definitely recommend checking out the open resources. But if you would like to, donate to our coffee fund, we don't make much money from this, but it does pay for my tea shop habit. Probably is about the limit of this. Please feel free to buy a version of the book. I'll briefly just mention a little bit about history. So DeepChem is a project that's been around, I think, seven years at this point. We've gone through a lot of evolution. We started life. The first version, I think, ran on Theano, which for those who've been doing machine learning, while is a very cool framework that's no longer quite around. And we've evolved through, TensorFlow, and increasingly, I think, in our latest release, we swapped over to using PyTorch as the full main backend, but we also have JAX support as something people are very interested in. So I think, part of what we've been trying to do in the last year at DeepChem is really production stabilize it. So we spent a lot of work effectively, hammering down weird edge case bugs. And I can test DeepChem is quite stable and safe to use in production settings. We have been using in production settings for some time. So if you're a big company, like Absci, that would like to actually use DeepChem in a production workflow, I think it's quite stable and ready to use, and I'll say a bit more about this at the end of the talk. Okay, so with that, that's just kind of a rough high-level introduction to what DeepChem is and what we do. But I thought it might be fun just to talk about some recent science that we've done, through DeepChem and through collaborations spurred by the open community around DeepChem. But before I kind of go into talking about some more recent scientific results, I'll just pause there in case anyone has any more questions or about DeepChem itself and the open community around it. Going once, going twice. Awesome. So let's talk about some science. So I think that there's a number of open questions I think around doing machine learning on molecules that have really pulled in a lot of attention I think over the last couple of years. One of the big ones, of course, has been using generative methods to design new molecules. This is of a lot of interest I think to people working on small molecule drug discovery, among other things. Like you could say, if you could have the computer kind of dream up a molecule, and that could potentially be a powerful source of finding innovation that you or your scientists may not have thought of. Now, there are a lot of challenges to doing something like this, and I'll say a bit more about them. But this is a project that kind of grew out of some open source hacking about one of our kind of contributors, Nathan, was finishing up his PhD, and he had gotten excited in kind of working on small molecule generative methods, but it wasn't what his thesis was focused on. So he kind of ended up working with us a little bit over the summer, and we ended up writing a paper together actually about some of the results that we achieved. The core idea behind kind of some of this early hacking that Nathan started was that there's a couple of new innovations that came out of both the machine learning and computational chemistry ecosystem that we were excited by. The first is normalizing flows, I think, are a tool that really has been popularized by DeepMind, among others. It's a way of transforming a simple probability distribution, like the little Gaussian you see on the lower left-hand side, into a very complex probability distribution, like the complex distribution you see, on the left-hand side for the Gaussian and the right-hand side for the complex distribution. And normalizing flow is a type of invertible transformation that satisfies certain mathematical conditions. For the mathematicians in the audience, it effectively ensures that the partition coefficient is 1, which means that you don't have to mess around with some painful challenges around estimating partition coefficients. And this means that you can kind of compute probabilities very nicely. The other kind of computational chemistry advance was that some really exciting work, I think, by Mario Kren out of Alana's Perguzik group was coming up with a new representation format for molecules as character strings called selfies. So one of the challenges in generating molecules is that typically the language that is commonly used to represent molecular structures is called smiles. And smiles have the kind of bad habit that you can write a syntactically valid smile string that is chemically meaningless. Like you can have too many bonds, you can have like valences that are broken. So selfies took the step of trying to tie the grammar of the string closely to the structure of the molecule. So in theory, if something is a syntactically valid selfie string, it should be a molecule that can exist, at least in principle. And I think the basic idea behind this paper is thinking, well, what if we just glued these two methods together? By using selfies, we can kind of ensure that we always come out, if we can sample a valid string with a valid molecule, and then we can use normalizing flows as a way of kind of learning the very complex distribution over molecular space that is occupied by real-world molecules, and use that to kind of pull out complex molecular samples. And here's kind of a brief top-line table. Like this very simple idea actually turns out to work quite well. So there are kind of sophisticated methods out there for sampling molecular structures. A lot of the way these methods work is they kind of assemble a molecule, atom by atom. So you can kind of view the molecules as like a graph, like nodes and edges, and you kind of stick on a new atom one at a time into a place on the graph. And this tends to work well, but it tends to be quite complex to code up and maintain. And it also is very complex. Whereas our method has the approach of just being like, oh, just take two methods that are already kind of well known elsewhere, glue them together, and bam, you have something that's almost as good as the state-of-the-art technique in terms of sampling valid, unique molecules. And I think one of the things that we were able to do because of the simplicity of this method, we were able to slot this, I think, into just a simple synthetic active learning pipeline. So the idea behind an active learning pipeline is you can have kind of an automated scientist where you're able to propose new experiments, in this case, new molecules to take a look at. Then you can use some downstream method of evaluating the goodness of these molecules. For the simple synthetic task we looked at, we just used a simple proxy metric. But in reality, this might be like an experimental or an assay evaluation or something else like that. And then you can take the results of this and then feed this back into the original sampler to generate your next set of hypotheses. So at a very high level, this type of active learning loop is like the same process we as human scientists do when we're trying to solve a problem, but it's trying to automate this into this like, iterative loop structure that, in theory can be faster and more efficient than a human. In practice, I think I always keep a human in the loop when I'm doing any real science. Like I don't trust any algorithm enough to like not actually have humans looking at it, but I think it can actually be a systematic way of like coming up with ideas that maybe you might not have thought of. Like we all have blind spots. And I think the algorithm has blind spots too, but it has different blind spots than a human. So I think it's a very powerful assistant to have in the tool, to have in the room when you're doing scientific discovery challenges. And here's just a brief result from the paper, and kind of the money shot is probably D, where it's sorry, the core result is probably in D, where we show that in a simple synthetic challenge, this active learning kind of loop is able to, in 3 iterations, find the maximally interesting molecule on a small toy problem, whereas a simple random search takes, I think, up to like 35 steps of iterations, of experimental iterations to find the maximally interesting molecule. So you can maybe see in principle where if you're running, let me say, for you all, maybe you're trying to design an antibody. Instead of 35 experimental iterations, if you're able to do the same in 3, I think things start to look very interesting. So, I'll say also, of course, this is kind of a, toy synthetic baseline. I think that, we've been separately looking afterwards in ways of kind of fleshing out some of the science here. And I'll also give a shout out to Connor Coley's group, where Nathan ended up kind of working with partly after his time working with us in DeepChem. And I think they've been doing some amazing work on kind of extending active learning into drug discovery and building out frameworks for making that more reliable. So I think, and I'll have a citation at the end where I actually point you to the paper that we wrote up about this, and this appeared in, I think, the NeurIPS Machine Learning for Molecules workshop 2020, I would like to say. So before I move on to kind of the next paper here, just pause in case anyone has any questions about that last work. Awesome. Going once. Great. So kind of continuing the theme about molecular generation, which is another project that we ended up working on, and this is a collaboration with researchers at CMU and Julia Computing, was looking at ways of generating new molecular structures using a new class of methods called score-based generative models. So here's another kind of diagram like the one I showed earlier, and I'll try to explain the difference between score-based methods and normalizing flows. So you might remember just a minute ago how I said in normalizing flows, you use a mathematical trick to ensure that the partition coefficient, which is effectively, for the non-mathematicians in the room, like if you have a probability distribution, all the probabilities have to add up to 1. Otherwise, like, saying something has a probability of 3 makes no sense. It can be like 0.25, that makes sense, but 3, that's not sensible. So what you do if you have a complex function that describes the probabilities of something, in the real world, oftentimes you might have this like awkward factor where when you add up all the possibilities, it comes out to 5 instead of 1. And you might say, well, let me just divide everything through by 5 and call it a day. And that 5 is what's typically called Z, the partition coefficient. So it turns out that estimating this value Z is extraordinarily complex in practice. It's it's been a while since complexity theory class, but I would like to say it's something like P-space complete or something horrendous like that. So normalizing flows use a clever mathematical trick to effectively ensure that you always maintain Z equals 1. But this means that you have to constrain the type of transformations you can perform. And in practice, what that means is you need very deep networks before you can actually sample very complex distributions. Whereas with score-based models, you effectively apply an alternate trick where you say, well, instead of me trying to learn the probability distribution outright, what if I just try to match the gradient? like I try to say that instead, let me effectively take the derivative of the log probability. And this turns out if you do a little bit of kind of math, if you can picture like a function divided through by z, and you take the log of it, then you're subtracting log of z instead. And then you take the derivative and that constant term vanishes. So I apologize for kind of doing math in words without writing anything down, but high level idea, score matching means that you can have more complex functions rather than the constrained functions of a normalizing flow to model a sophisticated probability distribution. And it sidesteps the difficulty with having to kind of estimate this partition coefficient. So we were excited to see whether you could adapt these score-based methods to generating molecules. And we also brought in kind of our friend selfies that is just really cool advance. So we were like, okay, let's just do it on selfies so we don't have to worry about some of these syntactic issues. So when we started this project, as far as we were aware, a lot of score-based methods had only been applied primarily to generating images and generating, not even much to generating text, but we're curious to see whether we could apply it to generate molecules. And effectively what it turns out was that we were able to get the system to generate valid molecules, but there were some interesting trade-offs where we effectively found that there were some limitations still that prevented the molecules generated from being entirely useful. Let me just pull up a little bit of a picture here. Effectively, so we were able to use these score-based methods to sample diverse and kind of interesting looking molecules. But one of the things we found is that the variance of these methods was very broad. So effectively, you couldn't tightly constrain the molecule sampled to a region of space that was close to the ones you cared about. For example, you often might say, have a few sample molecules that are active against a particular target. Then you actually want to stay close to that region of chemical space so you can look around it, but not say, look, next town over. Instead, you want to look next block over, as it were. And so this is actually something that these score-based methods still struggle with. And I think, this is an issue that other people in the literature have kind of wrestled with, where you have this sampling challenge where controlling kind of how these methods learn is still an open question in machine learning. So I think part of what was exciting about this project is we were able to get it to go somewhere interesting, but we also, at the end, kind of when we wrote up this this paper for a workshop, we did not have state-of-the-art results, which I think is also one of the cool things where like sometimes you have science that's in the fly. So we have continued working on this project, and I think we have better results, but don't yet have something that is quite state-of-the-art. It's kind of raw science. But again, I think that's the fun part of like a global effort like this, where again, I actually have not met any of my co-authors on this paper. Actually, on any of the papers I mentioned, I've never met them in person, but it's all kind of mediated through this like decentralized distributed community that's DeepChem. So I think with that, I'll kind of, swap over to discussing a pair of papers that I think I'm personally very excited about. But before I pause there and then move on, like any other questions about generative methods, either normalizing flows or score-based methods? Awesome. Okay, now let's learn about some large-scale pre-training. So, one challenge that, I've kind of seen working in machine learning on, molecules or really other scientific applications is that there's never that much data. And I'm sure for you all have faced similar challenges where if you're looking at scientific data, generating real data sets is costly. You need to run real experiments with real kind of reagents, real materials. It's not like the world in tech where, annotating an image is something that can be done at very rapid bulk scale, or you can fool your users into, clicking your CAPTCHA images and doing your annotation for you. So this means that, in doing scientific machine learning, we're always at a low data realm where there's always going to be less data than we would like for building out systems. So one of the, interesting things that, other folks and us started noting a few years ago, there's much larger unlabeled data sets where you say have raw chemical structures or raw, protein sequences or genomic sequences, then information about actual like experimentally measured quantities with them. So if you could somehow leverage, unsupervised or self-supervised learning to learn from this raw data without experimental labels, and learn something about the structure of small molecules or the structure of kind of protein or antibody sequences, there's some potentially really interesting things you can do here. So for us, we were interested particularly in small molecules. So I think we started looking at applying, basic transformers just to work directly off, Smiles representation in this case of small molecules. I'll briefly mention we did try selfies, but in this particular use case, it doesn't seem to make a big difference. Like the transformer seems to learn about it fine either way, and the smiles are actually faster to run at scale. So there's a little bit of a difference with kind of the two previous works I mentioned. And the basic idea here is that, what if you just train a standard transformer model using the same methods that people use in natural language processing on these small molecule data sets? And the core idea for how we train a transformer model on these data sets is we do what's called mass language modeling. Effectively, what you do is in a mass language model, if you have, say, like a sentence, you might, say, mask out certain words in the sentence and then train the model to infill the missing words. For a molecule, what this means is you effectively mask out certain atoms or bonds in the molecule, and you ask the algorithm to learn how to predict the missing atom or molecule. Imagine if you have, say, a molecule and you kind of pull out a carbon, you're like, okay, I have a blank that has 4 bonds coming out of it. What could it be? There's really, well, basically only one answer if you're looking at like organic molecules. So learning these types of, learning to solve these types of challenges, I think, teaches these models some basic understanding of chemistry. So what we did effectively was in kind of this first paper from a couple of years ago, we trained, I believe, on a set of 10 million molecules using this mass language modeling objective. Then we took the resulting model and we fine-tuned it on some downstream benchmark data sets from the MoleculeNet benchmark suite. And I think what we found was a little bit underwhelming. A basic random forest kind of pretty consistently outperformed this fine-tuned transformer. So, on the face of it, these results were like, well, why are you bothering to train an expensive transformer when a simple random forest can do better? But we did note this very interesting kind of like early scaling law. We did Appalachian studies where we kind of trained on subsets of the data. And the pattern we picked up was as we started training on larger collections of molecules, starting in this case, I think from 100,000 up to 10 million, we saw steady increases in downstream performance on tasks. And then mentally we said, well, what if we continue drawing the line further out? Like, can this reach a point where this actually can outperform standard methods for predicting the properties of molecules? And for those of you who followed advancements elsewhere in technology, you'll know this is kind of you look at GPT-3 or other things OpenAI is doing. If you'd get one of these transformers big enough, they start doing very magical things. So with this study, I think we ended up at the 10 million. Like with the student who was leading it, we kind of did a lot of this on Google Colab, so we left it there. But we did also take a little bit of a look at things like attention, where we're able to pull out estimates using the transformer of which regions of the molecule were contributing the most to downstream predictions of its properties. So I think the nice thing that this paper, like we kind of learned a little bit about like the scaling law, and we're also able to pull out some ways of analyzing what portions of molecules contributed meaningfully to downstream performance. And I think with that, we kind of wrapped that project up, but we did actually extend this work out into a sequel that we called Converta 2. So this project, picked up right where we left off with the previous one, where we started asking, well, can we go one step larger with how we train these things? And we also experimented, I think, with another a self-supervised task. So previously I mentioned this mass language modeling task where you block out certain tokens and have to infill them. But another task that we started using in this version is something called multitask regression. And this has been proposed a few places before in the literature. The idea is that for a molecule, you actually know a bag of basic properties about it already. You might know its molecular weight. You might know the number of rotatable bonds that it has. So, and these can be computed very simply with basic algorithms. So what if you trained the transformer model to learn to predict the molecular weight given the strength? This actually turns out to be actually a useful task, because it teaches it to kind of learn some basic knowledge about what the components of the molecule are and what contributes, what amount of mass. And if you train the transformers this way, I think this actually turns out to be quite a useful self-supervised objective. And this is a very rough diagram of the architecture that we built out. the idea here was that we used the mass language model pre-training and also the multitask regression pre-training. And then we fine-tune these on downstream tasks. One thing that, one trick we found, I'm not sure if I actually have a slide for this, but I'll just mention it. One trick we found was that training the multitask regression models was quite expensive. I think it was something like a week or two on like an A100 to get a multitask regression model training. But so we couldn't really do much hyperparameter tuning on the multitask regression. But we did a correlation study where we found that hyperparameters that worked well for the mass language modeling challenge also tended to work well for the multitask regression. So we're able to use the mass language modeling as a proxy task for selecting hyperparameters, which we then use on the multitask regression. And there were a couple other like tricks like this that we ended up using. I think we discussed them in the paper, but training large transformer models is definitely a work of art these days. It's not something where it's really out of the box. It's more like you have to learn kind of the magic knobs to make these things actually behave. But when we got all the magic knobs behaving, I think one thing we found was that when we scaled out and to larger collections of molecules and we added in kind of this new self-supervised task, well, we were able to get better results. We were able to now see that the converted to models are on average, I would say, able to outperform baselines on downstream tasks. So you do see the scaling law kind of carrying out. But there are, a few interesting points here where if you notice, sometimes it's not the biggest model that actually does the best. So sometimes the model that was trained on 10 million molecules or even 5 million tends to outperform the model that was trained on the full, 77 million that we considered in this paper. So there's a lot of mysteries around these models still yet. Like we now, in the last paper, we showed that there's an interesting scaling law, but this type of transformer-based free training didn't really beat downstream methods. And now I think we have a way to, achieve numerical results that are, potentially more predictive than other methods out there, like the ChemProp or other standard baselines. But we find that there's an instability in the training still. So I think that, to not spoil, but we have not solved this yet. I think there's still a lot of work being done. This is an active area of research for us. So, Converter 3 is slowly in the works and we're trying to get this wrapped up. But I think this is, if I had to say, this is an area where I think that, these large, we call it a chemical foundation model, I think is going to be fundamentally important in the science in this field. And I think we have some more cool results, but I think we've not yet anything that's ready to share. But we'll have some kind of more results kind of coming out of kind of DeepChem and other collaborator groups, I think, over the coming months. So let me, before I transition to the next part of the talk, let me just pause there in case anyone has questions about chemical transformers.
I guess I had a question, a more general question. So thank you for presenting a couple different frameworks and papers. And maybe your next section is already discussing this. So if it does, feel free to just move on. How does DeepChem as an open source software framework make it easier to do the previous 3 projects that you've outlined, as opposed to just, using bare bones PyTorch and, let's say, the standard ML libraries for serializing and storing the interim neural nets and doing checkpoints and so on?
No, that is a great question. So I'd say that there's kind of 2 parts to this, in that, like, I think that there's DeepChem, kind of the library, but there's also DeepChem, the open source community of researchers. So I think all the collaborations that were listed above were kind of done by people, I think, none of whom had ever met each other in person, and who, like, all these were catalyzed by people working in the broader framework of DeepChem. What I will say is that I think the first project has been fully integrated into DeepChem, so it's now within DeepChem 2.6. The latter 2 projects are still, like, active research, so they're actually not, production released into DeepChem itself. So it's more that these projects are, as we make advances, we then package it out into the DeepChem releases and release it more broadly to the community. But it's not the case that I'd say we view DeepChem as a vehicle for taking some of these advances and spreading them out and making it easy for other people to use. But I think that DeepChem as it stands today it's still like one limitation we have is that I think that DeepChem is a powerful tool for packaging and making easy a model to use once it's kind of baked. But I think it's not a lower level library like PyTorch or JAX, which focuses on making it easy to experiment with different models. So we do a lot of our raw hacking in PyTorch and JAX ourselves, and then we wrap that up into DeepChem in a nice shrink wrapped kind of unit that makes it easy for people to use. So I think the goal of DeepChem here is to make it easy for people to use these advances as they come out. But it is DeepChem as a framework, we're trying to still evolve into making it a tool for scientific experimentation as opposed to a tool for using some of these methods out there. I don't know if that fully answers.
Yeah, that does. Thank you.
I think, and this maybe actually ties a little bit into the future of DeepChem. So I think broadly, one of the big trends that, problems we've been trying to solve with DeepChem is that, yeah, how do we make this framework more composable? Kind of how do we make it more extensible? And, this has been a major engineering challenge. So I think I alluded to this briefly, but the Python ecosystem has changed choice of machine learning library du jour, like at least 4 times, each of which has kind of resulted in having to do like a rewrite of a major portion of DeepChem. This is a major challenge, like developing and maintaining a large open source library in Python is not easy. If you notice, there's a reason Google and Facebook and some other big organizations, like if you look at React or TensorFlow or PyTorch, all of these are backed by megacorp. There's actually, I think we're actually one of the larger, more indie open source libraries out there. Like NumPy is also a notable example. They've managed to get some more broad industry funding for NumPy and Pandas, which is great. But I think for DeepChem, smaller project, I think Deep Forest and a couple other companies have contributed in, but we're still trying to figure out how do we do the industrial scale engineering required to make this happen. And we have some cool ideas. I think this is, if you're curious, join the discussion on our forums. If you go to like forum.deepchem.io, there's several research posts about ways we can evolve kind of the future of DeepChem. And some ideas that have come out of like discussions on the forums and among our community is like DeepChem as a framework has been broadly evolving beyond chemistry. So we had last year like a really smart summer student who actually worked on solving partial differential equations within DeepChem. So he's added support for using what's called a physics-inspired neural net to solve some classes of PDEs using DeepChem. And for those of you who are kind of applied scientists in PD, I think neural PDE solution is just an amazing field that's blowing up. Solving high-dimensional partial differential equations has been very, very hard, I think, since the dawn of computing. It's still very, very hard, but I think there's now new tools that are making it easier to solve complex partial differential equations than ever before. And I think we'll start to see these tools trickling out more broadly to the community over the coming years. And we have some early tutorials and tools in DeepChem and plans for improvement over the next several releases, but PDEs are really hard. Another area where there's been, I think, a big kind of growth in DeepChem is people starting to use it for material science applications. So we have a small sub-community of people doing materials discovery with DeepChem. And there's a few tutorials and resources out there. And I think we're starting to work on an early draft of a manuscript kind of introducing the use of DeepChem for material science. And, I'll just mention very briefly, like the community has broadly been, building out tools towards broader science. So, the new code name for all the efforts is like the Deep Science Suite, where there's a suite of tools that are broadly applicable throughout the sciences. But again, I think a lot of our core efforts really work around small molecules and a little bit, and proteins and genomic sequences, I think, are always close to heart. So I'll just, very briefly, I'll come back to this guy in just a sec, but I'll kind of give a brief shout out. So, the company that I run called Deep Forest Sciences, we, help people, among other things, use tools like DeepChem within industry. So we built this, I think, very cool framework called Chiron that it has powered at its core by DeepChem infrastructure that, enables us to use some of the capabilities just of like, the cloud, but also I think some really exciting new algorithms and structures we built up. So we've been partnering with companies, mostly in biotech, a couple of people doing things in energy and other related fields to do some really cool things here. So I think that part of our goal with the Chiron system is to make it easier than ever for people, trying to do scientific discovery to leverage some of these cutting edge tools. And I think part of the, what I find really exciting about like the spectrum between deep chem or really deep science and deep forest sciences is that we're able to kind of have this like range from the entirely open, purely community driven to something that's actually enterprise grade and can be used in like a large drug discovery setting. And being able to span that spectrum and pick the right range. Like there are some customers that are not comfortable having anything being out in open, and we're like, yeah, it's fine. That's why we exist as Deep Forest Sciences. Then there are some, like for example, we have some collaborators at national labs that it's quite hard to get that governmental bureaucracy through, but we're like, oh, it's open source, and they're like, great, we'll download it off GitHub and give it a try. So just that gives us a range of flexibility that I think is part of what makes us interesting. And just quickly to go back here, this is maybe a partial answer to your question, Dennis, which is about how does this become more composable? This is a kind of a quite difficult software challenge. If you look at TensorFlow, TensorFlow 1 was amazing. It was rigid, but it did what it does really well. And then they tried to make TensorFlow 2, which was going on YouTube, so I'll be careful about what I say. But I will say they faced the fundamental challenge of trying to rewire, change out the engine on a car that worked well. And the new car, it still runs, but everyone kind of moved to PyTorch. And I think that it's because it's very hard to kind of take a system that actually works well and make it kind of broader and more capable. So this is something we've been wrestling with in DeepChem, which is that we would like to enable, as you said, make it easier for people to do scientific discovery through DeepChem, rather than just making DeepChem a vehicle for taking new algorithms out there. And we have some, several iterations on prototypes. I think we're starting to get better at this, but this is one of the core, this is a very hard software engineering challenge that we're trying to avoid the pitfalls that some large projects in the past have made here by. And I think one of the things we commit to is that we don't want to break anyone's code. So if you depend on DeepChem will be production stable. We have a very long deprecation cycle. So we're trying to kind of move in a measured fashion, but I think we are making steady progress. And I think in a few years, this will be something that is a tool for discovery and innovation, in addition to being a tool for kind of taking discoveries out there. But yeah, I think with that, just kind of going a little bit out of order today, but thank you for kind of inviting me. That's about all the material I had prepared. But if anyone has questions or things they'd like to discuss, happy to.
Yeah, thank you so much for the wonderful presentation, and it's actually right on time. We have some more time for questions. So just as a reminder, if you have a question, you can either press the raised hand button at the bottom of your screen, and then we'll call on you real time with audio. Or if you prefer, you could just type any questions you have in the Q&A window, and I'll just read them out for you. And if you think of questions after today's presentation, you can also always reach out to Bharath or myself directly. With that, I'll just give people 30 seconds to see if we have any questions.
So while we're waiting, I'll also mention that we have been trying to get better support for antibodies, I think, into DeepChem. I was hoping we could get a Google Summer of Code student this summer, but we didn't have any. We had a number of, we'll have a number of excellent students working with us, but no antibody student for the summer. So if you know any smart students who'd like to help us improve our antibody support, we do proteins pretty well actually, but in general, but I'll just put a shout out there in case anyone wants not to get anyone in trouble, but an evening fun project that is all open source, how we could use your help. Okay.
So it looks like we don't have any follow-on questions. Once again, thank you to our speaker, Bharath Ramsundar, for presenting to us today. And thanks to everyone who also joined. I hope everybody have a great rest of your day, and keep an eye out for future editions of our Absci Invites seminar series.
Awesome. And thank you again for inviting me, Dennis. And thank you everyone for listening along, and feel free to reach out to me or Dennis, as you mentioned, anytime if you have questions.
Thank you, Bharath. Bye-bye.